As Austin said, really at Abstrax, we're interested in cannabis as well as hops now. But really, this talk's going to be focusing on the phytochemistry that we've been kind of uncovering within cannabis, especially with respect to aroma. And so, just to give you a quick overview about kind of what Abstrax is as a company and what we're interested in, we're primarily, a research institution, I'd say, driven by discovering new flavor and fragrance sort of compounds that we then manufacture for various industries. This involves hops and cannabis, particularly cannabis at first, and now really moving forward with the hop chemistry. We do cannabis extraction in Long Beach. We have a fully licensed lab there with a constant intake of new material that we can be analyzing. We do purification and then obviously analysis that we'll be talking about today. But a big part of what I think makes Abstrax so unique and different from so many different corporations in general is our leadership is really big on education and dissemination of the chemistry that we're trying to learn. And so, this for me, as somebody who just loves to do research, I feel like I have a creative mind. The fact that we have an outlet to tell people about what we're discovering is just really awesome for myself. These are some of the brands within the cannabis industry and the brewing industry that we've worked with over the years. And I'm sure this has grown. I think this is a year or two old. And in the R&D department, the research and development department, what are we really interested in? What are we doing? And what is the path forward once we've discovered all this new chemistry? And so really this all starts with targeting flavor development. We want to understand how certain compounds come together to make a unique aromatic character or a flavor profile. And again, that could be in cannabis, hops, and other botanicals beyond that. And so that involves a lot of chemistry. It involves compound discovery and synthesis. It involves extraction optimization for our products that come out of Long Beach. Moving really excited about this, moving into kind of the biological and toxicology of these compounds and this chemistry that we're discovering. And then we're really moving forward towards using all this data that we're acquiring, using these advanced new models that are coming out or algorithms, that everybody I'm sure is aware of in this room, to make sense of this and do something with this data. And we have done some cultivation studies as well. So we're touching all facets of chemistry, I feel, and that's really unique within the cannabis industry in particular. And really, I think what's so unique about cannabis as well, just broadly, is this plant that so many people are interested in for many, many reasons. It touches almost all of the different kinds of kind of aspects of chemistry. So analytical chemistry, you have potency analysis for the cannabinoid content, the aroma analysis, which is what we'll be talking about today, heavy metals for safety, pesticide analysis, all these sort of things. But then on the organic side, if we discover new compounds, these might be new natural products in nature, or at the very least, maybe new natural products in cannabis. And so that will require, structure solution from mass spec, NMR, et cetera, as well as figuring out, synthetic pathways to actually produce those in a lab. Biologically, I mean, this is the part I think a lot of people are interested in, is like, why does cannabis get people high? How does that work? Do all these other compounds influence that property of it? And there's just so much unknown in this, quadrant right here, I would say. But it's all very relevant, I feel, to this plant, and probably the most interesting thing that we're working towards. And then lastly, physical chemistry, extraction optimization, emulsions for beverages that people can buy now in Minnesota bars and stuff like that. And then product development as far as these extracts go, and then the hardware associated with actually extracting this. And so I just think it's really fascinating how cannabis touches so many things, and each of these play a part in understanding the plant. So I like to show this diagram because, again, this talk is about the chemistry of cannabis. And this is a Sankey diagram that kind of breaks down the chemistry of cannabis in a pretty intuitive way. And so if we think about the plant being here, you can break it down into these kind of general sort of characteristic classes. And so the major class is going to be your cannabinoids that many of you are probably familiar with. It biosynthesizes THCA, CBDA, all these acid forms of these cannabinoids that when you smoke it or you produce an extract, you can decarboxylate, and then that's actually what gets you the psychoactive effects. But our talk's really interested in the aroma compounds, these volatiles that give cannabis its unique aroma. And so for so long, everybody's been focusing on terpenes in this industry and even in the academic sense. And so these include these classes such as monoterpenes, the monoterpenoids, sesquiterpenes, and sesquiterpenoids. And so there's probably about 40 or so that people really focus on within the cannabis industry, and have for a long time. But throughout our research, it became very evident very early on that these are not the whole part of what we're smelling. And so today we're going to be focusing on this tiny sliver right here. What we group everything else that's essentially not a terpene as a flavorant. And we do this because the chemical diversity and the functionality of all this chemistry is so wide that it's just easier to, especially within the cannabis community where there's not a lot of, education, if you will, as far as chemistry goes. It's easier to group these sort of as this general sort of class. And there's other stuff that we're going to be publishing on, hopefully in the next year. But the first story I want to tell you is about kind of uncovering what was for a long time, I feel probably the biggest mystery in the cannabis industry, which was what makes cannabis smell skunky. And so cannabis can produce a wide range of aromas. So you can see that these are some varieties that we had come through our lab, just took pictures of, and some of the major descriptors that some of these were given. You'll notice that this term gassy comes up pretty frequently, as does skunky. And so those terms are so ubiquitous with this plant, yet there's no indication that terpenes were the chemistry behind that. And so we know this because as we do flavor development, we have every single one of these, as well as probably 100 more terpenes on the shelf. And we can go in and smell these one by one to see what is their aromatic character. And so the one that a lot of people ascribe to be the scent of skunky cannabis for a long time, and to this day, I still see it on social media, is beta-myrcene. And I think that's primarily because it's typically higher in OG varietals, so like OG Kush, which was kind of one of the first quintessential skunky or gassy aromas. So, it's in the highest concentration. People just associate, okay, more intense or gassy, skunky aroma, higher levels on my COA, it's got to be that. But the truth is, if you have that in the lab, you smell it, it doesn't smell anything like that. The same for these compounds. None of these have the aroma descriptors that we were looking for as to that skunky gas-like aroma. And so when we were, trying to just hypothesize what might this be, if you just look into nature, you find very quickly that some of the most potent aromas in nature are the sulfur-containing species. And so in garlic, it's really well known for species such as diallyl sulfide, diallyl disulfide, the trisulfides in there. And all of these are really what make garlic have its really pleasant aroma when you're cooking. But if you think even more, intuitively, can it smell skunky? Well, what does a skunk produce? If you just look in literature, very quickly you'll see that it's a whole family of different thiols as well as just sulfur-containing VSCs. And so this is one of the major ones, which ironically, the only difference between it and one of the major components in cannabis is a double bond right here. But this is our hypothesis. We've said, well, if it smells skunky and skunks produce things with sulfur, let's hone in on that. And so what we did is we constructed a custom-built two-dimensional gas chromatography instrument. And so we, on top of having better separation and chromatographically, we also had 3 detectors working in tandem. We had a mass spectrometer, so this is useful for identification of the compounds. We had an FID. This is good for quantitation over wide dynamic ranges. And then really the key for this study was the sulfur chemiluminescence detector. And this detector is very specific. It only gives you peaks essentially if your compound has sulfur in it. So if you think that your sample has very low concentration analytes that contain sulfur, this is extremely useful, especially in complex matrices like cannabis. And so this is just an image of what our oven looked like at the time. If any of you work with GC, you probably have a heart attack because of all these columns going around in here. And so, getting this up and running was a struggle, I'd say. But once we kind of got going, the results came in pretty quickly. So to explain to you how GC cross GC works, 2D GC, I have this kind of brief schematic. So we can think of the cannabis aroma as being a very complex mixture of many, many compounds that when you open a jar that has this, you smell and release, and that's what you're actually detecting with your nose. In traditional GC, so one-dimensional GC, you separate these essentially by compound size or boiling point. So your smaller compounds elute earlier, they get larger, you get things, and eventually you might even get cannabinoids. And so you get a chromatogram that looks something like this, where you have intensity on the y-axis, and then retention time on the x-axis. So in GC cross GC, what you do is you first separate based on molecular size, but then you do a second separation based on polarity. And so you separate this way, and then you pull them apart in the second dimension. And what you end up getting instead are basically these heat maps, where you can think of these as like a topographical map of the Earth or something, where these mountains, these giant peaks, are higher intensity compounds. And so what you can see is that behind this row of peaks that arrive kind of at the bottom, there's quite a bit of chemical diversity up here with more polarity that you might not see if you're just running traditional gas chromatography. And so in combination with our detectors and then this separatory power, that's really what made it work for us. So to kind of explain from the instrument standpoint, how does this actually work? In essence, it's fairly similar to 1D GC, where you have your sample injector, you have a flow, we use helium, and then you pass through this first column. This first column might be about 30 meters long. I think that's what ours is. And then you hit what's called a flow modulator in our case. And this modulator basically groups every 7 seconds or so all the LUNs coming off of the column, and then it re-injects onto your polar column very rapidly. So this is maybe only that 7 seconds that it passes through this column and then goes to your detector. And so you're essentially taking packets of the chemistry that's not been separated by polarity and then separating it by polarity using this flow modulator. And so what does this actually look like when we're hunting these compounds down in cannabis? And so this is what it looks like on the SCD. You have this 2D chromatogram where you have a retention time of polarity essentially, and then boiling point or compound size. And then on the bottom we have the mass spec data. And you'll notice that there's a ton of peaks in this chromatogram. And of course, that's because the mass spec detects everything. It's not really specific in that way. On the other hand, the SCD has a handful of peaks. And so this sample was probably one of the most intense smelling skunky varieties that we had. You can see that all these peaks are pretty large. It's VSC 30, it's VSC 7, and they're really easy to pick out here. And so that makes it easy to go down here and then identify, well, what is that specific peak in the mass spec to identify? But that's all well and good if you have the chemical data. You really need, though, at the end of the day, we're modeling basically what are all the humans in the room saying this smells like? And so we're basically doing a correlation between the chemistry and that response. So we had a sensory panel get together and basically try to quantify how intense is that aroma, that skunky gassy aroma. And so we had about 13 varieties of different sample age, which for this doesn't really matter much, but the more important thing is we made them rank them based on olfactory score. In essence, what this means is that the higher that intensity is, give it a higher number. And so we have Baccio Gelato was the most intense aroma. And then at the very bottom, we had Blackjack, which is the lowest intensity aroma. And so we now have basically a metric or some data that we can try to correlate the chemistry to. And so if we look at the two extremes, the Baccio, the most intense aroma, and the Blackjack, it becomes immediately obvious that there's differences in this chemical data. And so when I saw this, I was like, okay, we're definitely on the right track here. And if you plot the 1D view of the 2D data, so essentially this is just like a 1D chromatogram where this is intensity now, and you line all of the varieties up based on that odor priority or the olfactory score, the VSC 30 peak shown right here concomitantly decreases as a function of basically that score. So it's almost monotonic with what people are actually reporting. So this to us was great evidence that we're on the right track here. But there was more than just this compound. And so this is VSC3. This is prenylthiol. Some other people, other folks call it MBT, 321 MBT. But this is really easy to identify and then characterize because it's in the NIST structural database. And it's not a complex molecule. But the other VSCs that we found, they did not index to anything that made sense in the NIST structural database. But luckily, if you just kind of briefly glance at the mass spectra, you'll notice patterns arising between the known compound and the unknowns. And what becomes immediately obvious is that, in particular, ions 41 and then 69 show up in each of these, very consistently. And really the only thing that's changing is the molecular ion. So the 116 here, which is just this guy with a methyl group stuck on it. This guy's 170, and it's the disulfide. And then, the diprenyl sulfide. And then the disulfide, the 202, is shown here. And then this one's really interesting because it's kind of got a bit more functionality than the others. And the 144 corresponds to the acetate. So we had these chemically synthesized, confirmed them with our GC, and verified that they're all being correctly reported. So once we did that, we were interested in saying, well, did these correlate at all with any specific sort of terpenes? And so two of the terpenes that folks talk about so frequently in cannabis, and especially the skunky aroma, is beta-myrcene and D-limonene. And so I've just ordered essentially this data as a function of decreasing prenylthiol content. And we will notice this beta-myrcene in particular shows no correlation whatsoever. And so I still don't think there's a great correlation between that dominant terpene and these prenylated thiols. You can have high D-limonene with very gassy aromas. You can have high caryophyllene. You can have high myrcene. It doesn't really matter. Cannabis and those sort of classifications all sort of produce, a multitude of levels that are detectable. One of the things that I definitely thought was kind of interesting was the chemistry of these VSCs is remarkably similar to that in garlic. And so the garlic VSCs are, this allyl group is essentially the same as the prenyl group. The only difference being you don't have the two methyls on the end. So we kind of just have a more lipophilic sort of molecule over here. But as you guys are probably aware, that chemistry drastically changes the aroma, right? Nobody would smell garlic and say, yeah, that smells like weed. And nobody would probably smell a ton of cannabis and say that smells like garlic, although that does happen occasionally for other reasons. But I think what's so fascinating about this is that garlic, all these compounds contribute to garlic's beneficial sort of, health benefits when people say, yeah, incorporate into your diet, those sort of things. And they can help things ranging from like, I saw like the one's an HDAC inhibitor, so that can be useful potentially for, cancer therapy, that sort of thing. And then the disulfide and then the trisulfide in the case of the garlic compounds actually can help in blood pressure. And they actually get converted, I believe it's into H2S in the body, which is like a vasodilator. And so it can actually lower your blood pressure. And so that's kind of one of the proposed mechanisms for why garlic can actually help in that regard. So the question is, cannabis produces something very similar, just a little bit more lipophilic. Does it also have similar sort of characteristics? They're in low concentration in cannabis, so you're going to fight that battle. But in a lab, right, in a biology lab, could you screen these against certain cell lines, that sort of thing, to see do they have some sort of functionality in that regard? The other thing that we became interested in is once we discovered these compounds, and we're coining this phrase cannabis-sulfur compounds due to their importance in the aroma of cannabis and that characteristic smell, but also that group is going to grow, as I'm going to show in some later slides. But we became interested in knowing how do these actually increase or change as a function of time during plant growth. So we worked with Ryan Pobans, our cultivation manager at the time. These are some pictures from the grow to see what happens. What we found is that in essence, during the flowering stage, so this is week 1 of flowering, so veg is already done, the photoperiods have been changed, etc. What we find is that there's minimal increase that we can detect for almost all of these VSCs up until about week 7. And at week 7, you basically start getting this rapid increase towards the end of flowering. And as the scientist in me is wondering, well, what if you didn't trim it here or start curing here and just let it grow out? What would have happened, right? I think that's an interesting study that we should do or somebody should do in the future. But we did end up cutting this down. I think it was after week 11. And so, that final data point after we dried them out was the highest. And then after only a week of curing, that compound concentration dropped pretty significantly. And so, when it comes to freshness, that people might use this aroma as a quality indicator, this sort of data and then how that changes over time even further, I think could have some pretty important implications as far as, kind of standardization throughout the industry for, shelf life and that sort of thing. I mentioned we do cannabis extraction in Long Beach. And so this is, a vape pen with the oil that we created, just as an example. But yeah, these compounds are definitely found in extracts. So if you produce these the right way, so we use butane, hydrocarbons, in order to extract these, you can definitely detect them. And so you can see that all of these are in these various butane hash oil samples. So they would look something like this in varied amounts. And interestingly, this VSC5, which is very similar in aroma character to the prenylthiol, I would actually argue that this guy is a little bit more skunky than the prenylthiol as its kind of main characteristic. But it shows up in all these, as do the other prenylated VSCs. So if somebody wants to use these as an indicator for quality in vape form, they can also do that as well. So one of the other things that became interesting after we found all this chemistry is, well, how does this kind of fit into the broader context of just the whole cannabis industry and the varieties that are out there? And this paper came out pretty soon after ours in 2022 from Nick Jikomis, who was at Leafly at the time. And they did this massive, like 20,000 data point analysis from testing labs across the US. And they essentially did, some really cool, statistical analysis. And they came up with this really nice plot that kind of showed that commercial cannabis, just looking at the dominant terpenes that testing labs report, essentially partition into these two main classes. You have these down here that kind of run together, this caryophyllene limonene and then myrcene pinene sort of cluster. And then you have this cluster up here that's terpinolene myrcene rich. And what's really interesting about this is that our data that he didn't know at the time maps directly onto this, wherein these prenylated VSCs are in much lower concentration, if not absent, in the terpinolene rich varieties. And we've seen that to this day. Whereas on the other hand, many varieties that kind of fit over into this, terpene profiles, if you will, they're very, very plentiful. And so I think that opens up a lot of interesting questions that maybe some plant scientists, such as yourselves, can answer, which is why is there's this relationship between terpinolene-rich varieties of cannabis and then everything else. And so I think there's a lot of potential, I don't want to say low-hanging fruit, but there's definitely some fruit to be learned here as far as the genetics behind why this might be the case. So to conclude this part, we identified this new class of compounds correlating to this aromatic character that everybody was looking for. And they are in very, very low concentration, but they have very high odor impact. We got a lot of cool press on this. So it was pretty fun. And this is the publication, and I can send it to somebody to disperse if you guys haven't seen it as well. Okay, so the next part of this talk is, what about all the other smells that people talk about in cannabis? These exotic aromas. What is exotic cannabis in general? And so going back to our Sankey diagram, the last time, in the last part of the talk, we were basically just focused on a subset of compounds in this can-of-sulfur line right here. But how does everything else sort of partition out to give the wide diversity of aromas that cannabis produces? That was really our next question after we solved that first one. So to conceptualize this with the team, we came up with this kind of schematic to kind of partition cannabis into three main classes that ended up working really well for us. And to this day, we still kind of use this broad general classification scheme. In essence, cannabis can range from being very sweet. I know that's kind of counterintuitive to a lot of people who are not familiar with cannabis, but it's true. You can have very sweet varieties of cannabis. They might be creamy, they might be fruity, they might be citrus or papaya smelling. That's why some of those are named like that. You can then be very prototypical. These are kind of your older varieties, more legacy genetics, something like maybe a Jack Hare that's more terpinolene rich, or maybe OG Kush, which it has that gas, but it's primarily dominated by the terpenic aromas that are in that product. But modern cannabis also can be very, very savory. And I think this is fascinating because, everybody thinks of exotic cannabis, I think, as sweet, but there's this equal but opposite sort of aroma characteristic on the other side that is reminiscent of onions and garlic or chemicals, which, that's a weird one, but we'll explain. Actually, we know some of the chemistry behind that now. So the question is, what is the chemistry that might lead to this side of the aroma spectrum and then this side of the aroma spectrum over here? So to do this, we procured 31 different rosin samples from around Southern California. And so we're very lucky in Southern California that rosin is a very popular form of consumption. It's what I would say that a lot of cannabis connoisseurs really enjoy. So a lot of varieties are made specifically with the intention of pressing and forming rosin. And so you get a lot of this aromatic diversity in rosin that you might not get quite as much in flower. But for those of you who might not be familiar with what is rosin, It's essentially, it's an extract. But instead of using solvents to pull this off of your plant material, you can follow this sort of schematic here. So you can take fresh frozen cannabis, which is basically you chop the cannabis off of the stalk, you flash freeze it, and then you take it over into a bucket of water. In essence, like literally, people just have garbage cans and then paddles, like a canoe paddle, and you gently kind of break off the trichomes in this ice water. They get brittle and they fall off essentially, just through mechanical action. From there, you can filter those trichomes with different micron size filters, and eventually, some people will then take that, freeze dry them. The steps variable depending on who you are, and then typically they'll oftentimes then be pressed under gentle heat in order to form this kind of sort of semi-liquid, in many cases, final product. And so the reason why this is so useful for us from what we found is that the concentration is higher than raw flower, period. And so we're typically looking between, at the minimum 5%, on average, probably 8 to 9% in Southern California. The other thing is that because we produce this from a large amount of different samples, it's going to be kind of the aggregate of that crop. So it's a bit more homogeneous than measuring a flower here, a flower there, flower there. The other thing is, compared to other extracts, I'd argue that ice hash rosin in particular has minimal processing. So you're retaining that aromatic character of the plant when you chopped it down. And so again, at the end of the day, the whole point of this aroma chemistry is, well, how are humans interpreting this, right? How are people smelling these different samples, and how can we kind of quantify that to then look back at the chemistry? And so again, we constructed a sensory panel to rate the samples through their noses, just literally blind tested, all that sort of thing. And one of the things we asked them to do is just rank this from 0 to 100, to be 100 being as sweet as possible, the sweetest thing that you smell out of these samples, down to 0, which is essentially no sweetness. And even with this panel that it wasn't blown out to over 100 people or anything, we see pretty clear partitioning between the samples that we got. And this is good for us for a few reasons. One, it told us that, okay, our sampling of the materials we got from across the SoCal is definitely has a wide aromatic range to our sensory panelist. But the other thing that was interesting here is that a lot of people kind of attribute dominant terpenes to a specific aroma character. So D-limonene, people see lime, so they assume, okay, that's going to be very, very citrusy. That's not necessarily the case. The sweetest variety, sure, that's limonene rich, but so is the one that is the most savory. And so there's not really a good correlation between these dominant terpenes and that aromatic character, which again is kind of going against the grain of what everybody's saying within the industry. And so to look at what the sensory panel, how did these varieties compare to one another in a pairwise sort of fashion, we do see that there's some highly correlated varieties, such as this sample right here. So this is just a Pearson correlation table where low correlation is down here at 0. So their aromas are very different. High correlation would indicate these things smell very similar. And the reason why I like this is it shows that we do have diversity in a pairwise fashion. We have some that are very non-correlated. We have some that are fairly correlated. But one that I'm going to point out here that I think is interesting, and I'm going to get to in a second, is Gorilla Glue. For some reason, this one right here was highly correlated with the sample garlic cocktail number 7. Okay, and so the aroma characteristics of Gorilla Glue probably shouldn't match those based on what one would think Gorilla Glue is and what garlic cocktail are. And so I'm going to explain how the chemistry can actually explain why basically this is a false positive for Gorilla Glue being labeled incorrectly. And so just to highlight kind of what does the sensory panel, what did these results sort of look like with our lexicon shown here? And so these density plots essentially show like how frequently were these terms used in summation. And what you'll notice is the most sweet varieties get a lot of terms kind of biased to the sweet side, sweet, fruity, banana, etc. way over here. Not a whole lot of density on this side. Whereas the most savory varieties, you get much more density down here in these sort of savory sort of characteristics. And so with all of the sensory data, it became important for us to now look to see, well, how does the chemistry map onto this? And so to do that, again, it's important for us to not tentatively assign chemicals, especially if they're very aromatically important, but to do it rigorously. And so we constructed an 80 plus compound standard for this publication. And so this is just all the analytes that we used shown on our FID. So they range from small molecules up to a little bit bigger with varying degrees of polarity. And our methodology was optimized for direct liquid injection for this low analyte detection. And so this is what all the data looks like if you just kind of glance at it from 30,000 feet. It all looks kind of similar, right? You got some density kind of in the middle. You got all the sesquiterpenes over there. It's very consistent throughout all of this. And so if you're not paying attention to the detail, you might make the mistake of saying grape pie do-si-do, which is the sweetest variety, is chemically identical to GMO. But we know that our noses don't say that. And so what are the differences in the chemistry? So this is a correlation table now of all the analytes taken into account. So terpenes, flavorants, et cetera. And what you'll find, again, is this data is going to be heavily biased by your high concentration compounds. So, your terpenes, your sesquiterpenes, and whatnot. Grape pie do-si-do versus GMO has a very high correlation chemically, even though aromatically they're very different. So again, this data kind of validates that there's something else going on here that we're not taking into account or we're not paying attention to. And if you just extract out by chemical classes, the monoterpenes, terpenoids, the sesquiterpenoids, they look even more similar. And again, this is the only stuff that people are reporting in their testing labs. So again, you won't be able to, GMO is going to show up the same as this one over here, which is just not true. Whereas when you start looking at flavorants, you start to see a lot more chemical diversity. And so this is where things start to get really interesting. So throughout the study, we aggregated all these new compounds that we're indexing, getting standards for, confirming, and we tabulated this graphic right here to kind of highlight some of the key chemical functionalities that I think are kind of interesting. So this is why cannabis can be fruity or sweet. It's not the terpenes, it's these esters, which is well known in plant chemistry that esters can oftentimes be very fruity or sweet. You can have these fruity or floral sort of aromas. Methylanthranilate is your quintessential grape flavor, and like all grape sodas, there's probably a ton of that in there. You have these really unique compounds that we only see very sporadically, but they're there, these phenethyl isobutyrate and the propanoate. But I'm really going to focus on these two classes because I think chemically they're the most interesting, and from an odor standpoint, they're probably some of the most important. So this is the chemical naphthol group, which contains indole and then scatol, and then this tropical citrus group that contains these 3-mercaptohexyl compounds shown here. So starting with the chemical naphthol group, this one's so interesting because cannabis does not produce a lot of secondary metabolites that have this functionality, even though indole, and that structure is wrong. I'm sure you guys picked that out, right? There should be that bond right there. You guys saw that, right? All you chemists out there? Okay. So indole is, it's ubiquitous in nature. It's found in a ton of different, natural products in our body, or, metabolites. It's in tryptophan, it's in melatonin. It's the core functional group in a lot of psychedelic drugs like DMT and LSD, psilocybin. And then scatol is very similar. You just have a methyl group sitting right here. And what's interesting is we find definitely a relationship between these two and varieties that are much more savory, and oftentimes in these sort of chem named varieties. And so the reason why people probably haven't reported these in the past, or at least one of the reasons why, is because they're really hard to see. They're really low concentration, and they're also buried, especially scatol, behind the sesquiterpenes, which makes it on a 1D system even more difficult to identify. So if you just look at a linear scale of our FID data, it doesn't look like there's anything there. But if you turn up the contrast, you start to see these tiny smudges shown here, that if you zoom in and you turn it up even more, you can actually see there's pretty well-defined peaks that index very well to indole and then scatol. And in particular, scatol, I happened upon that just throughout reading, literature and whatnot over time. And it's a really interesting one because it actually derives its name from the fact that it's found in feces, mammalian feces. And so when you guys are walking by a porta potty at the fair or something from a distance and it smells kind of bad, there's a good probability that scatol is actually contributing to that scent. But when it's in the correct concentrations, it can kind of take on different nuances. And so it can range from floral to chemical, oftentimes in the case of cannabis. and it's used in perfuming as well because it has this sort of chameleon-like sort of aroma characteristic. But again, tiny, tiny peaks, huge odor impact for those varieties. Now, I mentioned that the Gorilla Glue and that garlic cookies number 7, or the garlic cocktail number 7, were ranked to have very similar aroma characteristics. The reason why this doesn't make sense is because Gorilla Glue is very, very, it's named as such because it has sort of a chemical or very gassy, is what people would often call it, aroma. So it shouldn't be tropical in any sort of sense. Garlic cocktail number 7 is the complete opposite. It is very tropical. The first thing everybody would rank is citrus or some sort of descriptor sort of in that realm. So why is the sensory saying that they're similar, but the chemistry is not? And what it turns out is the Gorilla Glue has all of these tropical VSCs that contribute these very, very unique and pungent aromas that oftentimes override other aromas. And really, what this told me is that when TJ went out and procured Gorilla Glue, he was a victim of what happens so often in the cannabis industry, and that is renaming of products. And in essence, this is validation that TJ got scammed. He did not pick up a Gorilla Glue. He picked up a completely different product. And so I think this is, validation or, at least some evidence that this very fine detailed chemistry could potentially be used to help kind of validate products, that are on the shelves and validate that they're labeled correctly, that sort of thing. So to conclude this section, again, we've really hammered this home that terpenes are minimally influencing the perceived exotic nature of cannabis, even though that's what everybody's been focused on. These really low concentration analytes, they combine together to produce all the unique nuances, and working out how that happens is still ongoing. And we know some of the high odor impact stuff like scaton and the tropical VSCs, but they're very kind of one note. But there's more than just that when it comes to the bouquet of cannabis. And so we did publish this in 2023 last year. And so if you guys are interested in that, again, I can pass all these the links on to Mark and Austin. So the last thing I want to talk about is genetically similar cannabis. So the previous study we did, we were doing a wide breadth. We wanted to know, what does commercial cannabis kind of look like, at least in SoCal, right? Which we're hoping would translate onto the broader market. But what if you get really, really narrow and look at just very, very, very genetically similar cannabis? And the reason why I think this is interesting is because the breeding process requires basically, the production of seeds, which can be thought of kind of as siblings. And so those will be basically as genetically tight as you could probably possibly get, besides, cloning in essence, right? But you're not going to produce a new variety doing that 90% of the time. So, I'm using this graphic where I know this is chemical space, but if we think about this as the genetic space, we're going to zoom in as tight as possible to see, okay, between a bunch of phenotypes from the same crossing, processed exactly the same way, how chemically diverse are they and how aromatically diverse are they as well? And so I'm sure many of you guys know, talk about phenotypes more than I can. But, for those that might be as familiar, phenotypes are the physical characteristics of the organism that are observable, that you can kind of see, you can smell, and then, they're influenced by the genetics and then the environment going on around it. And so in the cannabis world, they use this term called phenohunting, which, it's not a real word. This is a term made up by the cannabis industry. But really, they're just saying it's selective breeding, right? You're selecting based on these traits that you want to try to propagate. And you're trying to find the best thing for whatever you're looking for. And so some people might look for terpene profile, percentage, yield, cannabinoid content, structure. All of these things are most likely taken into account for the majority of breeders. But especially in the case of rosin, aroma ends up being probably one of the most important things people are breeding for, because again, connoisseurs like weird new stuff. And so, even if it's not the highest yielding, a lot of these rosin producers will keep varieties that have unique aroma characteristics. And so this is just like a schematic from our TOC for the paper we just published. You take a mom and a dad, you take these, have these unique characteristics, and you produce things that might smell quite different, even though they're just from the same parents. And once again, going back to relating this back to the human element, we were interested in saying, so we were working with 710 Labs. So they're a really big producer in California as well as other states. They're not in Washington. I just found out, but they make really good products. They have very unique aroma characteristics. And so they produced 5 phenotypes of the same process. Starburst 36 is what it's called. I call it SB36 now due to trademark from whatever the candy company is. But anyway, so yeah, don't name your varieties after brand names, for those of you who do this. But in essence, what we found is that 2 of these varieties, SB36 phenotype number 1 and then number 40, were aromatically much more similar to one another than the remaining 3. In our sensory data, basically you get this, very high correlation between number 40 and then number 1, and everything else is a little less correlated. It's kind of, just look, they're correlated. So, you'd expect them to smell somewhat similar, their phenotypes, but, they're not quite as much as those 2. So again, starting to look from the top down, well, what does the terpene profile look like? And they look darn near similar, right? And on top of that, one of the main aroma characteristics of this was tropical tangerine and orange. Yet beta-caryophyllene, which is spicy, is the dominant terpene. Whereas everybody in the cannabis industry would probably be like, oh, that variety has to be limonene rich because it's citrus. But remember, it's not that when it comes to a lot of the citrus notes. It's these tropical VSCs. And so again, very similar chemistry shown here amongst the terpenes. And if you look at it on a correlation table, they look very, very similar yet again. But what was really interesting here is that our flavorant table mapped almost identically onto the sensory characteristics. And so this is really cool because it was like a very clear partitioning with the chemistry of these low concentration analytes between this grouping, and then everything else was just kind of off on its own. And so during this work, we did identify a few new compounds. One of them was this 3-mercaptohexanoate, hexyl hexanoate. So it's got the hexyl side chain over here. And so once again, this family of these tropical VSCs is continuing to grow, and it will continue to grow as we report more results over the next year or two. Again, very, very low concentration. And so, this, even though this has sulfur, the structural change completely changes the aroma characteristics from these gassy, skunky aromas. So, sulfur just elevates everything, but in totally different ways depending on the chemical functionality. Another interesting thing that we found was that we found octanoic acid and then decanoic acid in some of these varieties. And what's interesting here is that myself included, some of the panelists ranked some of the varieties as having a cheese nuance. I actually called it Stilton because that's my favorite blue cheese for one of them. And it's not strong, but it's detectable to some of those panelists' noses. And so these compounds, these fatty acids, are ubiquitous in cheese in general. And so it's cool now that that cheese is a whole kind of class of cannabis varieties that people name and unto itself, that we now have a little bit of chemistry that we can maybe track to see, are these really driving that sort of aromatic quality or not? But another interesting thing that we found is that there's a clear partitioning of the concentration of indole between the varieties that folks ranked much less sweet, kind of more prototypical savory, and the 2 that were very highly tropical and citrus. And so I think this is really cool because basically what it shows is that there's a hard cut between this being present and being completely absent, which makes me wonder if perhaps indole could be a good biomarker during breeding programs or whatnot, if you're trying to kind of go after more savory varieties in general, as well as it might have chemotaxonomic sort of utility, whereas you're not grouping based on terpenes anymore, you're classifying based on key small concentration compounds such as this. So in this paper, we essentially, once again, for the 3rd time, we show terpenes minimally are really correlating with the sensory descriptors of the work of the samples. These really low concentration analytes are just driving everything. Even in the phenotypes, you can have aroma diversity that maps really well with the chemical diversity. And then, they're genetically similar, sure, but you might be able to actually start to partition your phenotypes in a chemical way versus just, using your nose for analytes that might be really hard to detect with your nose, but you can at least see with your instrumentation. So with that, that's my story. I'd like to thank a few people because this work would not be possible without all of them. So Twinkle Pariani, she's been working Abstrax for a few years. She helped a ton on the exotic paper, the second story I told, and then that final study was her first author publication. So Twinkle, Manny, huge help in the lab. He's working on his own paper right now, so hopefully that'll be published, early next year. Marcos Ojeda, he helped a ton on all these projects, but really the sulfur chemistry in the very beginning, Marcos helped to do. TJ, my boss, he's the one who helps invest in the equipment, kind of guides some of the projects. Kevin as well, our CSO, our entire Abstrax team, and then I have some QR codes if you guys want to try to access those. Sepsolve and Marks, they're our instrument manufacturers, and we need them more than anybody because instruments break, as I'm sure you are all aware, and so we'd be screwed without them. So I have to give them a shout out as well. And thank you for your time. And again, Austin, thanks for the invitation.